The DNA is a polymer of dash, which consists of a sugar, a phosphate group, and a dash. What is your answer? Write it down in the comment section below, all right? Now, let me give you another question. What is the complementary DNA base sequence to the DNA strand TGCC80? What is the complementary DNA sequence to the base strand TGCC80? I need you to write down your answer in the comment section below, all right? A genome contains the necessary information for the production of a molecule of RNA. Is it a genome? Is it a gene? Is it a nucleotide? Is it a genetic code? Is it a codon? So a dash contains that necessary information. I want you to also write your answers below. These many other questions we're going to explore today. More than 60 questions. These questions will help you get ready for your NCLEX, for your any board exams, for your MCAT as well, also for your college exams, Anatomy 1 and Anatomy 2 on genetics. So I want you to look at this video, take your pen and paper, write down the questions, think about them. Check the ones I say that they are wrong, know why they are wrong. It's very, very important for you to know. I prepared about 40 slides that will also guide us in discussing these questions. All right? So I need you to gear up. Watch this video from beginning to ending. Also, make sure you make comments. Leave comments for me. I really want to know which topic are you finding difficult in biology so that I'll be able to treat it in my next video. Also, leave a like, click the notification bell so that you'll be able to get any video I upload at when due. All right? So let's start this discussion by understand what genetics is about. So genetics is study of heredity and gene transfer, right? Now, it's all about what makes us who we are. How you talk, how you smile, how you walk. Everything is all about genetics. Transferring traits from one generation to another. All right? That being understood, I want us to go straight into what really matters because this is not teaching. This is just a review, a rapid review. So I want us to go into what you most probably will see in your exams. Now, what you need to know is that the DNA has something we call nucleotide. What is nucleotide? Nucleotide is made up of repeating units, or rather, the DNA is made up of repeating units. Let's jump into what matters. Now, I want you to understand what the DNA is. The DNA is a polymer of what? Nucleotides. It consists of three important parts, which is a sugar, a phosphate group, and a nitrogenous base. So it's standing on the nitrogenous base. So that's why it's called a base. And the phosphate is the backbone, holding it strongly. All right. So the sugar is always, something that is at the core of the DNA. All right. Now, the sugar for the DNA is known as what? The deoxyribose. It's different from the sugar for the RNA. For the RNA, it's known as what? The ribose. That's a very big difference. Another thing you also need to understand is that the DNA has the nitrogenous base, just like the RNA. So the nitrogenous basis is divided into two groups, which is the purines and the pyrimidines. These are components of the DNA, purines and the pyrimidines, and they merge with each other. The purines are double ring structures. I need you to understand what double ring structures. They are double ring. Like an example of purine is adenine and guanine. If you look at the diagram, you see this is one ring, this is another ring. This is one ring, this is another ring. When you are looking at the nitrogenous base, that is double ring structure. So the purines are double ring, while the pyrimidines are what? Single ring. I want you to be able to understand it in the exam. All right. Now, example of purines are adenine and guanine. It's both found in the DNA and in the RNA. Both of them are found in the DNA and the RNA. Now, an example of pyrimidine is the CU COTS, cytosine, uracil, and thiamine. Cytosine is found in both the DNA and in the RNA, but uracil is found only in the RNA, while thiamine is found only in the DNA. So if you see a ring structure containing uracil, know that it is RNA. If you see a ring structure containing the thiamine, know that it's definitely what? DNA. Now, let's discuss the base pairing, right? The purines pairs with the pyrimidines, correct? So example of purines, adenine and guanine, correct? Example of pyrimidines, it's COTS, right? Now, the adenine pairs with the thiamine. Adenine pairs with, for the DNA, adenine pairs with thiamine. Adenine pairs with thiamine. And the guanine pairs with what? Cytosine. You need to remember this. Adenine pairs with thymine, AT, and guanine pairs with what? Cytosine. And something also you need to understand is they are connected by hydrogen bond. The adenine and the thymine has 2 hydrogen bonds, while the guanine and the cytosine has 3 hydrogen bonds. This is important information you need to know for your multiple choice questions. These things are very, very important for you to understand because it enables DNA replication and repair, which we are still going to discuss in a minute. All right. Now, if you see, remember that question I gave you initially? The question was asking, what is the complementary DNA base sequence in the DNA strand TGCC80? If you look at the slide, you will see if one strand is TGCC80, the complementary strand should be what? ACGGTA. Why? Because thymine pairs with adenine, guanine pairs with what? Cytosine. Cytosine pairs with guanine. Cytosine pairs with another guanine. Adenine pairs with thymine, and thymine pairs with adenine. So it's as simple as that. That's how you get your question in the exam hall. All right? That is just how you get your question in the exam hall. Now let's look at the organization of the DNA. All right? So we have the genome, we have the chromosomes, we have the genes, we have the DNA strand, and we have the nucleotide. This is the organization. The genome make up the chromosomes make up the genes make up the DNA strands, which make up what? The nucleotides. So but you need to understand each of these things for you to be able to pick the answers in the hall. For example, there is this question that says, what is all of the DNA in a set of 23 chromosomes referred to as? The answer is the genome. If you look at your slide, you see that the genome is a set of genetic what? Materials, which contains all of the DNA in a set of the 23 chromosomes, right? So what is the chromosome then? The chromosome is organized DNA structure. And when you are talking about the gene, you are talking about the functional unit of the heredity. All right? Which contains the information for the production of the RNA molecule. Remember, we have to produce RNA molecule for us to be able to produce proteins, which I'm going to go to later. I'm just picking out the pointed examples where you need to pay attention to. Pointed questions, rather. Now, there's a question that says, a dash contains the necessary information for the production of molecules of RNA. What is the answer? Gene. It's there on the slide. All right? Okay. Now, let's isolate the genome and understand what genome is. Genome is more like the library, the genetic library, contains everything, all right? Now, in terms of chromosome pairs, it has 23 pairs of chromosomes. You can't forget that. 22 pairs of autosomes, 1 pair of what? Sex chromosome. Now, 23 pairs means it is 46, correct? So, which is a complete set in humans. They will ask you this question. Also, there are 3 billion pairs of nucleotides. 3 billion. So there's a question that says the human genome consists of approximately dash pairs of nucleotides. Options: 3 billion, 3 million, 300,000, 3,000. The answer is what? 3 billion. You can't forget this. And the protein coding is 1 to 2%. So 1 to 2% of this 3 billion, you understand, codes for proteins. Taking a recap on the RNA and the DNA, they are cousins. I call them chemical cousins. All right. If you look at the slide, I specifically isolated the 4 differences between them, which you should understand. I've explained this initially, but I am going back to it because of how important it is. In terms of sugar, the RNA is ribose, the DNA is deoxyribose. R for D for D. In terms of bases, the RNA uses what? Uracil, while the DNA uses what? Thiamine for its pyrimidine. Every other thing about them, the purines, the same thing. You understand? The purines, the same thing. But the cytosine and the pyrimidines, the same thing. But the difference is uracil and thiamine. Now, in terms of structure, the RNA is single-stranded, while the DNA is what? Double-stranded, D for D. In terms of stability, RNA is less stable, of course, because it's single-stranded, and it is shorter than the DNA. Also, it's very important for you to know what the gene is. In one of my classes, I was telling my students that, the gene is just like, the DNA is a very big library, which we've already understood. And inside the library, you see the gene in a particular place. You say, oh, I want to make, maybe you want to make a dish, right? there is this golden manual for all dishes, which is DNA. Now I want to make rice. You understand? So you go to a particular section that is rice. That is what the gene is. So the gene is a segment in the DNA which contains information to produce RNA protein. Right? So that particular food you want to produce, you go to that particular manual, that particular segment. Why? Because there are a lot of food for you to produce. Right? Now, the gene is like the human gene contains approximately 20,000 to 25,000 genes. Like the human genome, rather. 20,000 to 25,000 genes. What are the functions of these genes? That is what they will ask you in your exam. So genes determine traits through their protein products. They determine traits. It's very important for you to understand this. Let me give you an illustration about the DNA and the genes, what the difference is, right? Now, think about it. Maybe you want to cook a very special dish. Let's say jollof rice, Nigeria jollof rice, right? Now, the DNA contains menu for all dishes, recipe, everything for all dishes. But you specific about Nigerian jollof rice. So what do? You go to the DNA and look for the segment that talks about what? The Nigerian jollof rice. That segment is known as gene. So gene is the DNA segment that contains specific information to produce a specific RNA or protein. You should know that so that they don't confuse you in the exam hall, right? And genomes contains genes, right? Now, the genomes contains approximately 20,000 to 25. The human genome contains approximately 20 to 25,000 genes. And their function is to determine traits. Simple. If you know this, you are good to go. Next, I need you to understand the central dogma of molecular biology. It's a very simple thing, as the diagram tells you, that DNA moves to RNA, RNA moves to protein. It's as simple as that. So the DNA, there's always replication, right? I want to photocopy that page, that segment for jollof rice. I want to photocopy it. That is called replication, right? Now I photocopied it and I transferred it to the RNA. That's called what? Transcription. Now the RNA now uses it, goes to the kitchen, calls other ribosomes, and do what? Use it to produce the jollof rice. That is what? Protein. It's as simple as that. So DNA stores genetic information, RNA carries the genetic message, and protein performs the cellular function after being produced. This is the central dogma of molecular biology, meaning how information flows. It's as simple as that, from here to here, from here to here. Now there is this question. Copying genetic information from DNA to RNA is called dash. Using the information contained in messenger RNA to make a polypeptide protein is called dash. Translation, transcription, DNA replication and translation, DNA duplication and transcription, transcription and translation, DNA translation and RNA transcription. The truth of the matter is that when you are looking at the slide, you will see what transcription is about. So transcription is from DNA to what? RNA. Simple. And where does it happen? In the nucleus. Transcription is what? From DNA to RNA. Where does it happen? In the nucleus. So if you look at that option, there are two, only one that starts with transcription. Which is the first one? Intelligent elimination. So the answer is what? Transcription and what? Translation. Meaning that the DNA to RNA is called what? Transcription. And mRNA to protein is called what? Translation. It's as simple as that. Now, if you continue looking at the slide, you see that the transcription has initiation, elongation, and termination. Initiation, elongation, and termination. And it's very important for you to know these three pathways. Now, when you're talking about transcription, which is from DNA to RNA, there's initiation, which is the RNA polymerase binding to promoter region. Simple. Then elongation, the RNA nucleotides align with the DNA template. Then termination, the RNA strand grows and detaches. That's termination. This is under transcription. Let me explain it in simple parlance. You come to the where that big food menu is. You go to the gene, which is the section for Nigerian jollof rice. Do you carry or do you tear out the paper? No. What do? You snap it. Because you can't bring it out. You snap it. That is replication. You snap it. So when you snap it, you do what? You come and show it to the RNA. The RNA will bind to it because that copy is known as what? DNA template. It's a template of the original thing. So the RNA will come and do what? And we align with it, which is elongation. And after the RNA has copied that template, it will detach from it, which is termination. That is what transcription is about. It's all about the RNA coming to get the information from where? The DNA. It's all about me going to the library to, get the information from a photocopied version, which is the DNA template. And when I'm done copying that photocopied version, what do? I throw it away or I detach myself from it. That is termination. So that is initiation, elongation, and termination. And then we ask you, where is RNA polymerase used? It is used in initiation part of transcription. But where is DNA polymerase used? It is used for what? Replication. You need to understand these concepts, all right? Now, when you're talking about translation, remember that messenger RNA that came to copy that DNA template is RNA that came to copy the DNA template is called what? The messenger RNA. And where did it come to copy the template? In the nucleus. Now, it will leave the nucleus and come to where? The cytoplasm. So transcription happens in the nucleus and translation happens where? In the cytoplasm. You need to understand this concept. Now, for translation, what really happened? There's still initiation, elongation, and termination. You understand? You start cooking the food takes time, and the food gets done. That's termination. So what happens? For initiation, the messenger RNA binds to what? Ribosomes. It's already gotten the templates, the copy of the template. So it binds to ribosomes, right? Now the ribosomes now use the copies to make what? Proteins. Remember, in the process, the tRNA, that's the transfer RNA, does what? Gathers acids and proteins, gathers them, because that is the ingredients we are using to cook. So the tRNA, that's the transfer RNA, gathers the ingredients. But the messenger RNA go and copies the photocopy, or the picture of the original. That doesn't make sense. So we have three things involved, which is the mRNA, the tRNA, and what? The rRNA, which is the ribosomal RNA, which helps in gathering everything and cooking the food. All right. So you need to know the roles of mRNA, you know the roles of tRNA, and know the roles of rRNA. And all these things are involved in translation. Is DNA involved in translation? No. Is DNA involved in translation? No. Because there's a question they asked. They said, which of the following is not directly involved in translation? The option: A, DNA, B, mRNA, C, tRNA, D, rRNA, E, ribosomes. The answer is what? DNA. It has no business with translation. Its business is what? Transcription. Now, when you're talking about the genetic code is something we call, or I want to call, the nature cipher, right? What is cipher? Cipher is more like a system for encoding or encrypting information. That's what genetic code is about. Like there's this question that said, the genetic code is the link between dash and the dash that they represent. DNA molecule, 20 amino acids, DNA molecule, 65 amino acids, mRNA molecules, anticodons, 20 amino acids, mRNA codons, 65 amino acids, RNA codons, 20 amino acids. The answer is RNA codons, 20 amino acids. So codons are more like 3 nucleotide sequence, right? And they specify a certain amino acid. Like if you look at the left-hand side of this diagram, you see codon 1, codon 2, codon 3, codon 4 with their respective amino acids. You see that there are 3 sequences, 3 nucleotide sequences. All right? Okay. Another thing is that, codons are universal. So they are the same thing in most organisms. They degenerate later in life, the multiple codons can specify the same amino acids, and they are unambiguous, meaning that they are very, very specific and simple. Now, let's look at the code system, right? There's something we call base triplets. We have something called codons and anticodons, as I've been explaining. Now, the base triplets are just 3 consecutive nucleotides in a DNA. That's base. 3 consecutive nucleotides, 1, 2, 3. They form a base, which I already discussed in the last slide. Now we have codons, which are 3 nucleotide sequence in mRNA. So the 3 consecutive nucleotides in DNA is known as what? Base triplets. But when you're talking about codons, there are 3 nucleotide sequences in RNA, that's messenger RNA. When you're talking anticodons, they are complementary sequences in tRNA. You need to understand these things. You really need to understand these things. So base triplets is DNA, codons is mRNA, anticodons is what? tRNA. So you should know what they're asking. Now let me give you a question. One of the questions, they said, which molecule contains anticodons? What is your answer? tRNA. Because in the option you see a DNA molecule, a tRNA, an rRNA, a cDNA? The answer is what? A tRNA. Anticodon, tRNA. But codons is mRNA. While base triplets is DNA. I don't want to oversay this thing, right? So as a recap, the key players in translation is the mRNA, which carries the genetic information from the DNA, all right? The tRNA, which transfers specific amino acids to growing polypeptide. You must know the function. Then the final one is the rRNA, which forms the structures of the what? The ribosome. So these are the three, and they have three specific roles. You must know these roles. You must know these roles. There's something we call post-translation modification, right? Post-translation modification is talking about, how you've already produced post-translation. Translation is to produce protein. You've already produced protein. We want to modify that protein because if that protein is bad, it becomes pathologic. Remember, what we are dealing with is anatomy and physiology. And what is physiology? Function, function, function. The right function. We want to know the real things for us to be able to understand the unreal things. So when you are doing pathology, you're talking about the abnormal, but physiology is the normal. So after producing cooking food, you have to test it to know if it's good. That is what we call post-translation modification, right? If the spice is not enough, you add some added spice, to make it taste very nice. You have to modify it. After sewing a clothes, you have to remove the ropes. That's post-translational modification. There are 4 different things that usually happen. The one is folding. You understand? Number 2 is addition of, that's phosphorylation and glycosylation. That's chemical addition. Another one is cleavage, like cutting some proteins that are not good, cleavage, cutting off. Then if you cut off here, what do? You throw away this bad one and you join the good one. That is called what? Joining multiple polypeptides together. All right. So these are the 4 ways of post-translational modification. There is this question they asked. They said, after translation, a protein may undergo structural changes called dash modification. Post-translational. Post-translational. Because they give you options like the polyribosomal, secretory, post-transcriptional. You see? Post-transcriptional. Then they give you splicing. Please. The answer is post-translational modification. All right? Now, there is this question I saw that says, since there are no genes coding for carbohydrates, nucleic acids, or lipids, how do cells produce them? Remember, everything we've been discussing is about producing what? Protein. So, how does cell produce these things? If you look at my slide, you see that since there are no genes coding for carbohydrates, nucleic acid, or lipids, enzymes encoded by genes synthesize these products. That is the option you should be looking for. Because they will give you options like cells can produce the carbohydrate, nucleic acid, and lipid. Now, they can tell you cells import these products from others. It's a lie. They can tell you cells absorb this product by endocytosis. Fat lie. Then cells are born with all the products they need. Absolute lie. So the answer should be that enzymes are encoded by genes. Synthesizes this protein. All right? Because gene codes for enzymes. Of course. Gene codes for enzymes. Then the enzymes will now catalyze some synthetic pathway, as you are seeing on the diagram there. There are a lot of pathways there, but that's what I'm going to treat during biochemistry. Now, what happens? The enzymes now catalyze the production of some biomolecules, like what? Like carbohydrates, like lipids, like nucleic acid. So that solves that question about, how are carbohydrate, nucleic acid, and lipids produced? They're produced from enzymes encoded by genes, right? Now, I want us to also look at the cell cycle. The cell cycle is very important, extremely important, because when you do pharmacology of anti-cancer medications, you understand this cell cycle very well, right? And it's also very important in genetics. Now, I'll be very brief. The cell cycle has, different phases. We have the G1 phase, we have the S phase, we have the G2 phase, and we have the phase, right? The G1 phase, the S phase, the G2 phase, and the mitotic phase, which also contains the cytokinesis. I need you to follow me so that you'll be able to understand this thing properly. Now, the first part is the interphase, right? So the interphase contains the G1 phase, the S phase, and the G2 phase. The G1 phase and the S phase, then the G2 phase. I had to put it on the diagram. So if you look at it very well, you see that during the G1 phase, that's the question they will ask you, what happens? During the G1 phase, what happens? Cell growth protein synthesis, organelle replication. The S phase, DNA replication, chromosome duplication, and the G2 phase, the final preparation for mitosis. The question they will ask you will be, the G1, the S, and the G2 phases of the cell cycle are collectively called what? The answer is interphase. The options they can give you: anaphase, prophase, telophase, cytokinesis, this phase. Please, G1, S phase, G2 phase are what? Interphase. It's very, very important for you to know these things. And also, you need to be able to know what happens in these phases. The G1 phase, as I told you, is for cell growth, protein synthesis, and organelle replication. Everything is just changing, preparing. In my class, I give an illustration. For example, I have a big anatomy class, and because it's too big, everybody wants this class to be divided. What do? You go and tell the administration. That is what happens in G1 phase. Normal, everything is normal, right? But you go and tell the administration, oh, we want this anatomy class to be divided. What does the admin do? The admin will be like, okay, if you want it to be divided, first thing we have to do is to get a new professor. So it's no longer Dr. Nchebe, it's now another professor. You understand? Because another professor has to teach the second class that is going to be formed. So assuming I am the DNA, right? So we are trying to break the DNA. That is called DNA replication. Because this class, we have a DNA. This class has to have a DNA. So if going to be in this class, we need another professor to be in this class. I need you to remember that illustration. And after that, what happens? Oh, we have to find another teacher, another professor, right? Then, okay, the management now comes to the class and announces to the class, everybody get ready, prepare. We're going to break this class. That is G2 phase. So DNA replication is the S phase. G2 phase is where they came to tell everybody to prepare. So preparation for what? Mitosis. It makes sense? Okay. Now let's go to mitosis proper. But before we go to the mitosis, replication, DNA replication, I've already discussed it initially. There are 3 enzymes that I want you to remember. One is the enzyme that helps for unwinding, like zipping open, because it's double helix. You zip it open. So that unwinding is done by the helicase. After that, there will be synthesis, which is done by the DNA polymerase. Then after that, they join back, which is done by what? The DNA ligase. Remember I told you replication is like getting a template, snapping that particular segment. So it has to unzip for you to snap. It's as simple as that. So 3 important enzymes: helicase for opening the 2 strands up, and the DNA polymerase, right, helps to take a picture of that particular place we want to create that complementary strand. Then the original has to be joined back together, back to normal, which is the DNA ligase. Remember that. Extremely important. Okay, now that is S phase. This thing happened in S phase, right? Now let's jump into the phase. That is the mitotic phase, right? The phase has 4 parts of it. Remember, we are dealing with mitosis, not meiosis. So it has 4 parts of it. So the first one is the prophase, metaphase, anaphase, and telophase. How do you remember that? Pass anatomy test. Passed my anatomy test. So if you start with the I, that's interphase. That's SGS. Interphase. I passed my anatomy test. Then the last one is cytokinesis completely. I need you to remember this. Very, very important. Now, you need to know what each of them does. So we have the prophase, we have the metaphase, we have the anaphase, and we have the telophase. You must remember what each does. Let me continue my story. Okay. So we are preparing, right? So the last stage of the interface is the G2. That's the preparation. Now we continue that preparation in the prophase. So P for P, preparation. But you need to understand, what is that preparation? Chromosomes condense, chromatins shortens and thickens, right? Calling into compound rods. That is a question that they can ask you. That is the question they will ask you. So that's prophase. So prophase is more like advanced preparation. Everybody is now carrying their bag, coming around. Oh, we are celebrating. Everybody is reducing all their baggages. So chromosome shortening, chromatin shortening, and compact rod. That is for prophase. Remember it. Now, metaphase, meta for middle. Oh, everybody, come to the middle, come to the middle, come to the middle, come to the middle. Line up according to your heights. Let's divide this class. That is metaphase. So chromosomes align the equator, the middle, for middle, right? Then after that, what do we do? I'll say, okay, divide these people, all of you into two. You shift to this side, you shift to this side. So dividing the class, but remember, we are still in the same cell. The cell has not been divided, but you are dividing the class. That is called what? Anaphase. So sister chromatids, they move. They move to the different poles. That means move to the different class, but it's still in the same class, right? At the end of the day, we have telophase. Telophase is nuclear envelopes, reforms, chromatins, decondense. So when they move to the different extreme, by now, telophase, they are now at the extreme. Anaphase is dividing, telophase is they are now at the extreme. So all of them that carry their bag will drop their bag. So the condensing will decondense. Then after that, we build a wall, break the two classes, which is what? Cytokinesis. Cytokinesis is dividing the entire cell, cytoplasm, breaking the cytoplasm into two. So there are now two cells. I need you to use this story to remember this. You can never forget. So this is the prophase. This is the metaphase, lining up at the middle, aligning, and all those things. This is the anaphase. You see, there is a cleavage. They have now started going to their different poles. After that, we have the telophase. All of them are now in their different regions, dropping their bag. And cytokinesis divides this into two. Very, very important for you to understand. Cytokinesis divides it into two. There are a lot of questions we discuss on this, but that will be at the end of this discussion. All right. Now let me jump into karyotype. What is karyotype? There's a question on it. Karyotype is what? It's a portrait showing the 46 chromosomes. It's as simple as that. It's a portrait showing the 46 chromosomes. If they ask you karyotype, remember this in the exam. I'll give you a question on that. As a recap, somatic cells contains 46 chromosomes, 23 pairs in humans, known as diploid. That's 2N. Germ cells, 23 unpaired chromosomes in humans, known as haploid. All right? So other animals, they have their own numbers, which I'm not here to discuss. I'm here to discuss humans. Now let's talk about alleles. Allele is a form of a gene, right? It helps for, genetic diversification. Very, very important for genetic diversification. And remember, each individual has two pairs or two copies of each gene. That is what we know as alleles. If you look at the slide, you see what I'm talking about, that alleles are alternative forms of genes, right? We have dominant, we have recessive. The dominant are the ones that are expressed. The recessive are the ones that are usually masked by the dominant. But the recessive can also be expressed if they are together, right? So a recessive allele is expressed as a phenotype only when two copies are what? Present. But the dominant is expressed only if only one copy is even present. The dominant will still be expressed. That's very, very important for you to know. And there's something we call heterozygous and homozygous. Heterozygous is when it's one recessive and one dominant. But if it's two recessive or two dominant, it is called homozygous. So a genotype is a genetic makeup of an organism, right? Consisting of alleles inherited from parents. Does it make sense? It's a genetic makeup of an organism consisting of alleles inherited from parents. It helps to determine potential traits, but most times or sometimes it's not always dictated in appearance. That is genotype. It's very, very important for you to know. That's when you say, oh, this is AA, this is AS, this is SS. But when you're talking about phenotype, phenotype is the physical observable expressions of a genotype. Physical observable expressions of a genotype. Usually influenced by both genetics and also by the environment. All right? In terms of patterns of inheritance, we have simple dominance, we have codominance, we have incomplete dominance. So simple dominance is usually talking about, when simple dominance is talking about when a dominant allele marks the recessive allele. It's as simple as that, right? You just mask it. When you're talking about codominance, the two of them are heterozygous and they fully expressed. There are two different dominants and they are fully expressed, like the AB blood group. Two of them are fully expressed. That's codominance. And when you are talking about incomplete dominance, it's just like getting a hybrid, right? Just like your hair color, getting a hybrid. One parent can have white hair, another person will have dark hair. Then their children will have intermediate hair colors. That's incomplete dominance. It's very important for you to just understand this concept. I'll give you questions. Now, there's something we call polygenic inheritance. Polygenic, many gene. Polygenic, many gene, leading to what? One feature of phenotype. All right? So multiple genes, different locations, forms one thing. For example, the eye color, the skin color, the intelligence, height, all these things are formed by different genes. That is what we call polygenic inheritance. I'll give you an example. Sex-linked inheritance refers to genes located on this, the sex chromosomes, like the X and the Y, right? Sex chromosomes. So, like these genes, they follow unique inheritance patterns because the X and Y and the XX have different chromosome composition. But what you need to know is that most sex-linked traits are usually affecting the male because the male, they have only one X. So once that X is faulty, it affects them. But women can, they have double X that can, neutralize the effect of that one S. That's the idea. So female usually need two of the recessive alleles for them to, manifest any sex-linked trait. But usually female are the carriers because it's a female that give an X to the male. So the mother will pass an X to the son. So a carrier mother always leads to a sex-linked trait for the son, not for the daughter. All right. So examples of sex-linked traits, we have hemophilia, we have Duchenne muscular dystrophy, we have that affects the dystrophin, we have the red-green color blindness, right? Very, very important for you to know what sex-linked trait is. We're going to treat questions on it. So if you look at the board, you see the Punnett square. So the Punnett square for sickle cell, for example, you see, this is the genotype ASAS. Their gametes, they're bringing out their gametes A, S, A, and S. This is Punnett square. This is the first AS. This is the second AS. A matches with A to give AA. A matches with this S to give AS. S is with A to give AS. S matches with A to give SS. So meaning that the outcome, we have one normal, that's AA. We have two carriers, that's AS, and we have one sufferer, that's SS. So if you are to calculate this physiologically, phenotypically, you see that AA and AS, they're actually normal phenotypically. They look normal. So it will be 75% over 25, right? But if this is genetic calculation, it will be 25%, 50%, because AS and AS, 50% they're carriers, and genetically they are different from AA, and 25% SS. So you have to understand the genotypic calculation and the phenotypic calculation. Phenotypic calculation, oh, this person is normal, oh, this person is not normal. So 75%, 25%. Now, genotypic calculation, oh, this person is normal, this person is a carrier, and this person is sufferer. So 25%, 50%, and 25%. This is how to arrange the Punnett square. This is what the Punnett square is about. All right? Now, the basis of population genetics, we have gene pool, we have allele frequency, we have Hadley-Weinberg's equilibrium equation. Now, the gene pool is talking about the alleles for every member of the population. The allele frequency is talking about the proportion of a specific allele, in the gene pool. So if there is a specific allele that is occurring, that is the allele frequency. Now, we are using a Weinberg equilibrium to predict an allele frequency. I will not go into this here, but just know that this is p squared 2pq plus q squared equals to 1. What are we calculating? The p squared is the frequency of homozygous dominant genotype. The 2pq is the frequency of heterozygous genotype. And the q squared is the frequency of the heterozygous recessive genotype. So you have to be able to put all these things in their respective places for you to be able to use this equation. I wish that I would say a equation that I can make this calculation for you. When they ask you about mutation, tell them that mutation is a permanent change in the DNA sequence. Simple. There are different types of mutation, right? There are point mutations, there's friendship mutation, there is inversion, there is duplication, there is translocation. And what could cause mutation? Radiation, chemicals, and replication errors. Right? I don't want to stress it. So just go look up all these types of mutation because they can ask you in your exam. All right? But I'll give you some questions. Mutation can be harmful, it can be neutral, it can be very beneficial. Right? Harmful, neutral, beneficial, which is very, very rare. So this genetic testing, like karyotype analysis, we usually use it for, to detect chromosomal abnormalities. We have DNA sequencing, which identifies specific gene mutations. We have different types of tests, that's like diagnostic, predictive, parental screening. All these things helps you, to identify things, which is why genetic testing is extremely important. And this genetic testing, we helps us to understand genetic engineering, which is trying to reshape everything we know in life, right? Like if you look at the diagram properly, that's CRISPR, that's 9. It's a recombinant DNA, right? Like combining DNA from different sources. When you want to combine DNA from different sources, you join them together, then you use the CRISPR to do what? To edit. And after editing, what do? You can modify these genes to use them to treat diseases. That's what gene therapy is about. So recombinant DNA, CRISPR-Cas9, and gene therapy. Extremely important for you to know. In the clinic, we can, genetic disorders, we can use them to treat, we can use genetics to treat genetic disorders, right? Different treatment approaches, enzyme replacement, targeted drugs, gene therapy, very, very important, and also counseling. So let's take some questions. Very, very important. I've already discussed a whole host of questions, but now I want to also discuss some questions on the cell cycle so that we'll be able to understand each other very properly. As a recap to what we've discussed. Now, there's this question that says, okay, I've asked this question before, G1 S phase and G2, that's the interphase phase. We all know what happens at the interphase phase. Very important. That's called the resting phase. All right. Now, we have this question that says, cytokinesis overlaps with which phase of mitosis? Which phase overlaps with cytokinesis? The answer is telophase, right? Why is the answer telophase? Because in telophase, remember, they are going to the extremes, going to the pole. And as they're going to the pole, this one divides in the middle. There becomes a cleavage. That is cytokinesis, right? So bear that in mind. Very, very important. That question is something that's always constant on your board. The stage at which chromosomes aggregate along the equator of a cell is dash. I want you to pause and attempt the answer. A, interphase. B, telophase. C, anaphase. D, metaphase. And E, prophase. The answer is what? Metaphase. Remember, I told you, when they come to the middle, everybody wants to divide. Everybody comes to the middle and line up according to your height. That is for middle for metaphase. You can't fail this question. Which of these processes occurs during a cell's first gap phase? That's G1 phase. What happens in G1 phase? All these questions, pause and attempt them. 1. The chromatin condenses into visible chromosomes. 2. The cell synthesizes proteins and grows. 3. The cell synthesizes the enzymes that controls mitosis. 4. The nuclear membrane or nuclear envelope breaks down. DNA is semi-conservatively replicated. So the first answer talks about DNA is semi-conservatively replicated, but all of us know that happens in the S phase, correct? That's DNA replication. The second one talks about the nuclear envelope breaks down. This happens in the prophase, usually, right? Then the third one says the cell synthesizes the enzymes that controls mitosis. It has to control mitosis, so it has to be produced before mitosis starts. This happens in what? The G2 phase. Now, the one says the chromatin condenses into visible chromosomes. We know where it happens. That is in the prophase. Now, one talks about the cell synthesizes proteins and grows. Yes. That's what happens in the G1 phase. So the G1 phase, everything is normal. Everything is growing. Everybody's just there in the class. So the cell synthesizes proteins and grows. That is G1 phase. Now there is this question that says, sister chromatids are pulled to the opposite poles of a cell during dash of a cell cycle. Okay, the options: metaphase, prophase, anaphase, the G2 phase, and the S phase. What is the answer from here? Usually we know that going to the poles is telophase, correct? But there's no telophase in this equation. So what else are we thinking about? Anaphase, because that's when, okay, everybody move towards the poles, right? Telophase is when they get to the poles. Then before cytokinesis, where they break. So here, the answer is what? Anaphase. Anaphase. It can be metaphase. Metaphase is the middle. All of them are lining according to their height. It can be prophase. Prophase is the condensing, preparing, and all those things. It can be G2. It can be S. That's DNA replication. So the answer is what? anaphase. You can't fail this question. Now, they asked a follow-up question that chromatin shortens and thickens, calling into compact rods during dash of mitosis. Do you remember what happened in the prophase? What did I say? I said the the chromosomes condense, chromatin shortens and thickens, calling into compact rods. That is the answer. That is the answer. All right? Because the question is chromatin shortens and thickens, calling into compact rods during dash of mitosis. The answer is prophase. Prophase. They are preparing. It is not interphase. It is not anaphase. It's not telophase, and it is not metaphase. All right? Now they ask this question: which of the following omits some stages of cell cycle? So the options: first one, interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. It's correct. It has all the cell cycle stages. So that can be the answer because they're asking which one omits. Always pay attention to the answer. Now, another option says G1, DNA replication, that's S, G2, the sequence follows, then mitosis, that's it. No, that's it. Oh, yeah. This is correct. This is correct. Yes, this is correct. Then the next one says interphase, mitosis. The mitosis, it follows. The next one says G1, S, G2, mitosis, it follows. Now, this last option says prophase, metaphase, anaphase, telophase, cytokinesis. Is it correct? No. Why? Because there is no interface. So the truth of the matter is that if you just know I passed my anatomy test completely, that's all you need. And remember, the cytokinesis is also known, it's also part of the what? The mitosis phase. So if they tell you interphase and mitosis, they are correct. Because interphase is G1S and G2. The mitosis is pass my anatomy test completely. All right? So you need to memorize this. You need to say it a lot of times for you to be able to understand this. Now, the question says, a karyotype shows the dash chromosome sorted and isolated from a cell in dash. How many chromosomes is karyotype? I mentioned it before, 46. And they are isolated where? In the metaphase. That's where you bring everybody and you isolate them. You want to count how many people are there. You want to arrange how many people are here. Everybody's aligning up according to their height. Did you see the karyotype? Everybody is arranged according to their height. Make sense? So remember that about karyotype. Now, a question says, if one allele is not phenotypically expressed in the presence of another, we said that it is what? It is what? Recessive. It's as simple as that. I don't want to waste time on this. But no recessive, no dominant, no heterozygous, and no homozygous. I told you, homozygous is like when you have two alleles that are the same thing, like small a, or big A, right? That's homozygous. They are homozygous, the same alleles. Now we have heterozygous, which maybe like capital A, small a, right? Or AS. That's heterozygous. They are not the same thing, right? Now we have recessive. Recessive is like when you have capital A and small a, or capital S and small s, or capital D small of the same thing. That's recessive. Dominant is when, so recessive is the smaller one. This one is small letter. Dominant is the one in the capital letter. So like A, capital letter A and small letter S. The small letter S is the recessive. The capital letter A is the dominant. You have to know these things, these terminologies. Like in this question, they are talking about if one allele is not phenotypically expressed in the presence of another, we say it is what? Recessive. But if it's expressed in the presence of another, we say it was dominant. And if the two of them are expressed, we say it is what? Co-dominant. Another question they're asking is, eye color is caused by genes at multiple loci. The phenomenon is called what? Pleiotropy? Pleiotropy? No. Pleiotropy is when one gene causes different features. One gene causes different features. That's pleiotropy. It's opposite of polygenic inheritance. So polygenic inheritance is when eye color is caused by many genes. So many genes causes that particular phenotype. That's called what? Polygenic inheritance. Polygenic inheritance. If you look at this thing, there are a lot of polygenic inheritance like eye color, hair color, intelligence, height. All these things are polygenic inheritance. We have incomplete dominance. We have multiple carriers. We have sex linkage, which we've discussed. I mentioned pleiotropy because we didn't discuss it. But remember it now because it will come out in your questions. Germ cells have 23 chromosomes, 23 unpaired chromosomes, and are thus called haploid. So germ cells are 23 unpaired chromosomes in humans, haploid. Somatic cells are 46 chromosomes, that's 23 pairs in humans, diploid or diploid, right? So you have to bear in mind this answer so that you don't fail it. Question says, the allele for normal hemoglobin H is dominant to the allele for the sickle hemoglobin H, that's small h. A male and a female who are both heterozygous for sickle cell hemoglobin have a child. What is the chance that this child will have sickle cell hemoglobin? So for example, if I want to say instead of H, let me say A, right? Then instead of small h, let me say S. So they are asking, what is the chance that this child will have sickle cell hemoglobin? If you look at the Punnett square that I'm showing you right now, the Punnett square, what is the chance that this child is a sufferer? 25%. Because if you check, this is normal, 25%, this carrier 50%, and this is the SS is, and that is what they are asking. All right, 25%. So you need to know what the question. If the question is saying, what is the chance that a child will be a carrier? That is 50%. Because there are two AS that you get. What is the chance the child will be normal? That is 25%. Another question says, the allele possessed by all members of a population collectively are called dash. Answer, gene pool. Answer, gene pool, not genetic drift, not population DNA, not genetic frequency. Answer what? Gene pool. You should be able to know the difference between gene pool and allelic frequency, even genetic drift. Look it up. And also they had a Weinberg equilibrium. Now there is this question that says, which of the following is not found in the RNA? Not found in the RNA. All right? Options, uracil, thiamine, ribose, and guanine. And what did I tell you? I told you that the difference between RNA and DNA, you can categorize them into 4: sugar, base, structure, and stability. For RNA, ribose, DNA, deoxyribose. Bases, RNA, uracil, DNA, thiamine. Meaning that you cannot see thiamine. You can see uracil, but you cannot see thiamine. All right? It's extremely important because if you look at the options say thiamine, uracil, ribose. Yes, you can see ribose. Guanine, yes, you can see guanine. Uracil, yes, you can see uracil, but you can't see thiamine. This is the kind of question you will meet. Now, there's a question that says, a segment of DNA that codes for the production of a molecule of RNA is known as what? We have codon, anticodon, base triplet, promoter, and gene. Please, the answer is gene. A gene, is that segment of a DNA that codes for a production, a molecule of RNA. That page where you take the recipe from. It's very, very important for you to know it, right? Because I took time to define all these things like genome, chromosome, genes, DNA strands, nucleotide. Even I talked about the genome extensively, right? Before I even start talking about base pairing rules and all those things. So you really need to pay attention to do these things because they are the ones that will come out in your exam. Now, there is a question that says, which of the following statements is false about gene? Genes can be turned on or off, their products are needed or not. It's true. In a given gene cell, some genes are never turned on. True. Once a gene is turned on, it is activated in all cells. False. A hormone can turn a gene on or off. True. So they says, which of the following statement is false about gene? The answer is that a gene is turned on. That once a gene is turned on, it is activated in all cells. Right? Because that is false. Genes are turned on and off from day to day, even hour to hour as their products are needed. Right? I want to cook rice. I go and get and activate the particular page for rice. Right? Many genes are permanently turned off. A version of a trait that requires a homozygous genotype for expression is called a recessive allele. Remember that. Recessive allele. So because we have dominant, we have recessive gene, and we have dominant gene. But this is allele. Now let's look at these things. Like the percentage of a population with a given genotype that actually exhibits the predicted phenotype is called what? Penetrance. I need you to understand it. The percentage of a population with a given genotype, but they actually exhibit that predicted phenotype is called what? Penetrance. That's the ability for that gene to penetrate and to showcase itself. All right? And because of this penetrance, it's not everyone that has the dominant allele for a trait like will be expressed. Sometimes you have a dominant allele for a trait and it won't be expressed because of penetrance. Because environmental factors affect allele a lot, right? It takes both heredity and environment to make a phenotype. So genotype and environment makes up what? A phenotype. All right? So let's review the stages of mitosis. During dash, the chromosomes become visible and condensed, prophase. During dash, the double-stranded chromosomes line up along the equator, that's in the middle, metaphase. During dash, the sister chromatids of each chromosomes separate and move away from each other, anaphase. In dash, the chromosomes uncoil as the nuclear envelope starts to form, that is telophase. Because when they get to the pole, they do what? They uncoil. When they get to the pole, they decondense. That is telophase. All right? So it's also good for you to understand the steps completely because it's a recap. We have the growth phase, that's the G1 phase, S phase, and the G2 phase, that's preparation. Then we have the mitosis phase, which is the prophase, metaphase, anaphase, and telophase. And finally, we have the cytokinesis. So the G1, S, G2 phase is arresting. That is the interphase. You understand? So I passed my anatomy test completely. It's very good for you to remember and memorize this sequence. I need you to think about these and tell me which one is correct. Females can be carriers for a sex-linked recessive. Okay, hemophilia is caused by a sex-linked recessive allele. This means that dash. Females can be carriers but cannot get hemophilia. The gene is found on the Y chromosome. A male can give the allele to their sons but not to their daughters. True? Nah. A male who has hemophilia inherited the hemophilia from his father. Nah. A male who has hemophilia inherited the allele from his mother. Yes. Why? Because sex-linked traits are usually on the X chromosome, right? Yeah. So if a man has it, he definitely got it from the mom because the mom gave him the X chromosome. It's as simple as that. It's very straightforward. That's what we call X-linked trait or sex-linked traits. You should also be able to know the difference between somatic cells and germ cells, right? So the reproductive cells are germ cells, while the somatic cells are what? The non-reproductive cells. It's as simple as that. So all of the body's non-reproductive cells called dash usually have 23 pairs of chromosomes and are thus called dash cells. Answer, somatic cells, 23 pairs of chromosomes, diploid. Simple. Somatic cells, diploid. D. A DNA replication is called semi-conservative because dash. That's what semi-conservative is very, very important, right? So it means that each daughter DNA consists of one new helix and one old helix. So if this happens during replication, so if DNA replicates, that breaks into two, right? What happens? That daughter cell that was formed has one new strand and one old strand. That is the meaning of semi-conservative. You can't fail this because they'll give you options like each daughter cell receives one new helix and one old helix. No, they do not receive. Each daughter cell receives one new chromatid and one old chromatid. Each daughter cell consists of one new chromatid and one old chromatid. No. Each daughter cell consists of one new nucleotide and one old nucleotide. No. The answer is each daughter DNA consists of one new helix and one old helix. So this is the DNA. It breaks into two. This one has a new one to it. This one has a new one to it. That is semi-conservative. Simple. What is a mutation? A harmful change in DNA structure. B, a beneficial change in DNA structure. C, a change in the size or shape of a cell. D, a neutral change in DNA structure. And E, a change in DNA structure that might be beneficial, harmful, or neutral. What is your answer? The answer is a change in DNA structure, right? Permanent change. It can be beneficial, it can be harmful, and it can be what? Neutral, as we've discussed. And something you need to bear in mind is that there are different types, which point frame shift, inversion, duplication, translocation, and others. How do they happen? Radiation, chemicals, replication, errors. They can be harmful, they can be harmless, they can be beneficial, but the beneficial parts are always very rare. But that does not mean that there is no beneficial part. So in that option, the right answer is a change in DNA structure that might be beneficial, harmful, or neutral. Now they gave you this question: which of the following events does not occur in a prophase? Options: DNA condenses to form chromosomes. Does it? Yes. It happens there, so it can't be the answer. The nuclear envelope breaks down. Yes. It can't be the answer. The nucleus breaks down. The mitotic spindle begins to form. All this is a preparation. You understand? Before you go to the metaphase phase. Now, the last option says the chromosomes are replicated. Is it true? Nah. All right? So you have to bear that in mind. So there's this question about the structure of the chromosome. It says a mitotic spindle fibers attached to chromosome via special structures called what? Kinetochores. Kinetochores. Not centrosomes, not keratins, not esters, not centrioles. It is kinetochores. Bear that in mind. I have to pronounce it slowly so that you'll be able to remember. Very important for you to know that. They can also ask you a question. What term describes nuclear division? They tell you something like interphase. Is it nuclear division? No. Cytokinesis? No. Cell cycle? No. Mitosis? Yes. Nuclear division is what? Mitosis. Don't forget. Nuclear division is what? Mitosis. Extremely important for you to remember. They are always asking this question: during which phase do sister chromatids separate and start moving towards opposite poles of the cell? It is anaphase. It is anaphase. When you hear that word separate, it is anaphase. It's something you cannot forget. All right? You can never forget it. Where in the cell does transcription occurs? In the nucleus. Where does translation occurs? In the cytoplasm. And what is the product of transcription? Messenger RNA. You have to know the difference between messenger RNA, tRNA, and rRNA. tRNA is the one that brings the amino acids. Messenger RNA is the one that went to snap the base pair, the DNA strand. It's messenger RNA went to. That's why it is the product of mRNA. And what is the product of transcription? And what is product of translation? Proteins. And after that, it will undergo modification. Very important for you to put this in your mind. Also, when they ask you to arrange nitrogen, base, carbon, nucleotides, DNA, chromatin in order, this is the order. I don't have time to read the question, but this is the order. Carbon, nitrogen, like carbon, nitrogenous base, nucleotide, DNA, and chromatin. You see? Carbon and nitrogen. Now, carbon is a part of the makeup of nitrogenous bases. Nitrogenous bases make the nucleotide. The nucleotide makes the DNA. The DNA, when it's together, tied together with histone, it makes up chromatin. Remember that. That is the order. Because they will just explicit, they will put carbon here, they will put chromatin here. No. Remember, it is carbon, nitrogenous base, nucleotide, DNA, and what? Chromatin. So finally, the genetic code for a polypeptide made up of 51 amino acids would have a minimum of dash bases. It's a simple thing. Multiply it by 3 because it is 3 plates, 3. So 51 times 3 is 153, and you get your answer. Yeah, this is very interesting. Like, we are able to do a lot of questions. Remember, this does not substitute for your classworks. It doesn't substitute for your lecture slides. No. This is for a rapid review, for you to be able to recap a lot of things you've been able to do, you have been doing in class, and for you to know the best way to attempt these questions. Right? I'm trying to place a kind of reasoning in your head so that you don't have a problem in any questions with genetics and also the other topics I've discussed so far. I'll be doing tissues next. I really want you to brace up for it because we'll be discussing about 50 to 80 questions on tissues. All right? So that you'll be able to answer any questions on tissues. So please try so much to like this video. Subscribe if you've not subscribed. You have to subscribe. All right? Please. And also try so much to make a comment. I need you to leave topics that you're finding challenging in anatomy and physiology for me. All right? So this is for students that are preparing for anatomy and physiology exams, students that are preparing for NCLEX, students that are preparing for MCAT, students that are preparing for USMLE. All right? So feel free to leave a message for me in the comment section. If you like the video, tell me you like the video. All right? If there is something you want me to correct in the video, also express yourself. All right? Thank you so much.